Good afternoon, everyone. I'm Virda Hafiz, and it's a pleasure to welcome you all to session 5 of the 8th International Conference on Applied Zoology 2025. Today's session covers some really exciting themes like pathology, drug discovery, ecology, genetics, and cancer biology. These are the areas that bring together both laboratory science and field based discoveries. We have an amazing lineup of speakers with research that ranges from natural remedies for hypertension to the nanoparticle therapies, cancer vaccine innovations, and toxicological and ecological studies in aquatic system. So let's get started. While we move forward, I would like to welcome our very first presenter, which is from Chile, and his name is Dr. Patricio. He would be presenting today size overlap, null models for understanding zooplankton community in extreme environment, Salar di Surere.
Hello, it is the presentation entitled Size Overlap Neural Model for Understanding Zooplankton Community in Extreme Environment, Salar de Surire, Marinica Parinacota Region, 18 South Latitude, Chile, from Patricio de los Ríos Escalante, from Catholic University of Temuco, Chile. In this slide, I have mentioned my affiliation address. As introduction, zooplankton assemblage in Chilean lakes and lagoons is characterized by its marked endemism, low species number, and marked calanoid dominance due mainly oligotrophy and other stressor agents such as salinity and natural ultraviolet radiation exposure. The aim of the present study was to achieve first statistical ecology characterization of crustacean zooplankton communities in Salar de Surire, that is a complex of shallow pools associated in saline deposits in Andes Mountains in the extreme north of Chile using overlap, size overlap null models. This is material and method. The study site. The site is a saline deposit with small shallow lagoons located in Andes Mountains close to Bolivian boundary. This is the geographical location and altitude above sea levels. You can see a photograph of the study sites and a map with the localization of the study sites. Data collection, so plankton was collected, filtering 30 later samples at the screen with 100 micrometers of mesh size. Specimens were identified in according to literature, and it was measured the total length. As that analysis, the new model is size structure was applied for total length collected specimens, and it was applied to determine the non-random patterns in size overlap of species. The data of this analysis were consistent of a matrix which each species was a row and each side was a column. Entries in the side was represented in the mean size of length of each species. The original matrix was then reshuffled for produce random patterns that would be expected in the absence of competitive interactions. The minimum difference and size uniform uh, were used. These neural models were performed using EcoSim R package and software R. In result, revealed the presence of size overlap in 5 of 6 models that in consequence revealed that the species reported shared ecological niche. In this site, we found only 4 species, the Cladocerans, Alona, and Macrotrix, and Calanoid copepod Bochella titicacae and Bochella calcaris. And in this table, you can see the mean size of each species. In this table, you can see the result that you can see the presence of size overlap in 5 models uh, with p value up, upper than 0.05. In this plate, you can see the graphs of respective new models that we use in this study, and this graph supports the result of the last table. As discussion, the obtained result will be similar to the description for other lakes around continental Chile. In this scenario, the zooplankton species reported worldwide ecological niche as phytoplankton glaciers and filters of particulate organic matter that will be similar to potential proposals for shallow lakes in Andean mountains in the north of Chile, as well as large and deep Patagonian lakes. In this slide are mentioned the acknowledgement, and in this slide are mentioned some relevant references for this study. Thank you for your attention.
It was pleasure having you, Dr. Patricio. Thank you so much for sharing your work with us. Next, we have Dr. Shumaila Mehdi from Government College University, Faisalabad, and she would be presenting today bedside to bedside approach, a novel natural remedy to treat hypertension. Let's welcome Dr. Shumaila Mehdi from GCUF.
Assalamu alaikum. This is me, Shumaila Mehdi, Assistant Professor in Department of Pharmacology, Faculty of Pharmaceutical Sciences, Government College University, Faisalabad. The topic of my presentation is bedside to bedside approach and a novel natural remedy to treat the hypertension. This one is the abstract of my presentation. What is hypertension? Hypertension refers to sustained high arterial blood pressure over time and is a major contributor to cardiovascular diseases and often progresses to severe life-threatening conditions such as myocardial infarction, coronary artery disease, congestive heart failure, renal failure, and stroke. Hypertension causes approximately 9.8 million deaths every year worldwide, and non-compliance is alarming. That is 50 to 70% of patients do not take medicine according to the prescription, according to the World Health Organization. So this trend is rising in specifically in Pakistan. So there are different risk factors, including the genetic predilection, desk-bound lifestyle, and intake of high-fat diet. So high-fat diet lead towards an array of aberrations, including the dyslipidemia, hyperinsulinemia that lead toward the endothelial dysfunction and cause atherosclerosis, which specifically lead toward the vascular dysfunction and hypertension. Presently available antihypertensive drugs have limitations, so research for newer drug is defensible. Medicinal plants and their bioactive constituents are commonly used worldwide to control the hypertension and its complication due to their non-toxic nature, availability, and affordability. World Health Organization has recommended the use of traditional plants for the treatment of chronic and life-threatening diseases, including the hypertension. Berberis lycium royal, that is an outstanding species for Berberidaceae family, traditionally employed in Ayurvedic and Unani structures. Berberis lycium has advanced from folk treatment into the topic of pharmacological scrutiny, in particular for its ability roles in combating cardiovascular and metabolic conditions. Reported activities of Berberis lycium are antioxidant, antibacterial, antihyperlipidemic, antiviral, antidiabetic, anti-cancer, anti-inflammatory, and antimicrobial. So the objective of our study is to explore the chemical characterization of Berberis lycium and to investigate the antihypertensive and antihyperlipidemic effect of Berberis lycium in high-fat diet and L-NAM-induced hypertensive rats, and to study the effect of Berberis lycium on inflammatory cytokines level and oxidative stresses. Plant was collected from the Azad Kashmir and identified in the Government College University, the Department of Botany in Government College University, Faisalabad, and the extract was prepared by maceration and evaporation. The final filtrate will be air dried, and the respective percentage yield was calculated. After that, the phytochemical analysis was done, that total flavonoid content and total phenolic content of the plant material, and quantitative analysis was performed by performance or high pressure liquid chromatography. An FTIR analysis was performed. In vitro antioxidant assays include the DPPH radical scavenging assay and hydrogen peroxide scavenging activity. Study design of our trial was that include the two phases: induction phase and the treatment phase. Induction phase, having 6 group of rats were given the high-fat diet for 8 weeks, and after that a freshly made intraperitoneal injection of L-name, that is 185 micromole per kg, after being dissolved in citrate buffer at the concentration of 0.1 molar at the pH of 4.5, was administered to all groups except the normal control group of animals. And this one is the experimental dosing of the animals. That group 1 is the normal control, group 2 is a disease control, that is having the high fat diet plus the L-name injection, and group 3 is the standard control or positive control, having the atorvastatin, that is 10 mg per kg, and group 4, 5, and 6, they are treated groups having the low, medium, and high dose of the plant material. After that, the biochemical analysis, after the completion of the trial, the biochemical analysis for in vivo antihypertensive assessment was performed, and with the body weight, fluid and food intake, non-invasive blood pressure measurement, inflammatory cytokines level, adipocytokines level, by ELISA, LFTs, RFTs, oxidative stress markers, messenger RNA expression of the candidate genes, and histopathology of the organs was performed. And this one is the gene expression profiling by qRT-PCR, in which first the RNA was extracted from the samples. After that, RNA quantification was performed, and the DNA was synthesized from the RNA. Then primers was designed and then reconstituted, and gel electrophoresis was performed, and for the optimization of the primer, and then polymerase chain reaction was performed to get the results. These are the results: that total flavonoid content and total phenolic content in the Berberis lycium extract, and the in vitro antioxidant assays, and the result in the DPPA scavenging activity, IC50 value of 76.25, and hydrogen peroxide radical scavenging assay, and the result was that is 40.426 as compared to the standard drug. This is the HPLC analysis of Berberis lycium, that having the flavonoids, phenols, as well as the alkaloids. These are the name of the constituents: quercetin, gallic acid, vanillic acid, benzoic acid, chlorogenic acid, p-cumaric acid, m-cumaric acid, and synapic acid. And berberine was also confirmed in the list of compounds in the HPLC analysis of Berberis lycium. This one is the FTIR analysis of the plant in plant extract, which contain the different functional groups. This is the hematological analysis in which the hemoglobin level, red blood cells, white blood cells, and platelet count was recorded. This is impact of the aqueous methanolic extract of Berberis lycium on the body weight, as we can see, as after the 4th and the 6th week, the diseased animals have the more body weight is increased as compared to the normal control group of animals, while the treated group of animals show the less body weight as compared to the disease control group of animals. This is the impact of the aqueous methanolic extract of the Berberis lycium on food and fluid intake, in which we can see, as after the 8th week of the study, the body food intake and fluid water intake was increased as compared to the normal control, while the treated animals show the less food and fluid intake as compared to the disease control group of the animals. This is the impact on the blood pressure. As we can see, the systolic blood pressure, mean blood pressure, and diastolic blood pressure, they are increased after the 8th week and as compared to the control group of animals, while the treated animals decrease the blood pressure as compared to the disease control group of the animals. Same is the case with the effect of aqueous methanolic extract of Berberis lycium on the hyperlipidemia, and that is triglyceride, total cholesterol, and LDL increased after completion at the induction of the hypertension, that as compared to the normal control group of animals, while the treated animals show the hypolipidemic effect of the extract as compared to the disease control group of animals, which have taken the high fat diet and the L-lean. While the impact of blame on the renal function test, that urea and creatinine and uric acid levels was increased in the disease control group of animals, and that is decreased by the treatment with the plant extract. Same is the case with the liver function test, that ALT, AST, bilirubin, alkaline phosphatase, bilirubin direct, and bilirubin total, they are modulated in the diseased animals, while the treated animals, they also improve the level as compared to the diseased animals. So effect on the inflammatory biomarkers, that is TNF-alpha and IL-6, they are increased in the diseased animal, and they are decreased as compared to the diseased animal with the treatment of the plant extract in the three different doses. An impact on the oxidative stress assays, CAT, SOD, and MDA. So the level were CAT superoxide dismutase and catalase activity was decreased in the disease animal as compared to the MDA level, that is increase in the disease animal, and that is reversed by the treatment with the plant extract. This is a qPCR analysis in which the cyclooxygenase 2, nuclear factor kappa B, endothelial nitric oxide, angiotensin converting enzyme, and perilipin, they are modulated with the treatment of the plant extract. This is a histopathological analysis in which the liver, kidney, heart, and aorta. The architecture is reversed with the treatment of the Berberis lycium extract as compared to the disease group of animals. This is the This one is the schematic flow diagram that summarizes the pathophysiology of the high-fat diet and L-NAM-induced hypertension and hyperlipidemia. It outlines the therapeutic impact of various doses of Berberis lycium tested across antioxidant and inflammatory and histopathological parameters. Conclusion of our study is the finding of the study indicate that Berberis lycium possess antihypertensive, antihyperlipidemic, antioxidant, and anti-inflammatory activities in high-fat diet and L-NAM-induced hypertension model. Through comprehensive biochemical, histopathological, and phytochemical analysis, the aqueous methanolic extract of Berberis lycium demonstrated a multifaceted therapeutic profile. So the extract progressed blood pressure, regulated lipid metabolism, modulated inflammatory markers, and restored antioxidant enzyme interest in hypertension and hyperlipidemic situations. So this study can be used for further evaluation of effectiveness of Berberis lycium in hypertension and hyperlipidemia in animals for its potential benefits. So this is all about my presentation, and thank you very much.
Thank you so much, Dr. Shmaila Mehdi. I would like to move forward. And our next presenter is Dr. Razia Noreen from Government College University, Faisalabad. She would be presenting today xanthine oxidase inhibiting and an anti-hyperuricemic effect of curcumin nanoparticles in monosodium urate crystal-induced bulby C mice model. Welcome, Dr. Razia Noreen.
Dear students, professors, and organizers, Assalamu alaikum. I would like to say thank you to the organizers of this 8th International Conference on Applied Zoology 2025. The topic of my presentation is xanthine oxidase inhibiting and hyperuricemic effect of curcumin nanoparticles in monosodium urate crystals induced valve C mice models. My name is Dr. Azia Doreen. I am professor in the Department of Biochemistry, Government College University, Rajasthan. And let's know about the hyperuricemia. Hyperuricemia is a medical condition which is characterized by an abnormally high level of uric acid in the blood. Uric acid is a waste product of urine's metabolism. And if we talk about the prevalence of hyperuricemia, 2.6% to 36% is present in worldwide and only 31.9% in Pakistan. It represents some symptoms like gout, kidney stones, trophy formation, urate, nephropathy, like here it has been shown in the image also. So for the need of the project, this is the study has been designed because of the multiple problems with the current therapies. The target of this study project was to find a therapy having following qualities, like which more efficacious than the present available treatments, least or no toxicity, cheap and easily available, and having least or no damage to liver and kidney. In this study, we have used the curcumin, which is present in the turmeric and contains a polyphenolic molecule. It has excellent pharmacological qualities such as anti-inflammatory, antioxidant, anti-cancer, and neuroprotective effect. So, however, its therapeutic value is restricted because of its quick metabolism, low bioavailability, and poor water solubility. The creation of nanoparticles is a potential way to get around these obstacles and improve its medicinal efficacy. So the objective of the study is the synthesis and characterization of nanoparticles of curcumin and then the evaluation of curcumin nanoparticles against the hyperuricemia in valve C mice model. In our treatment methods, first of all, the curcumin nanoparticles have been synthesized and then for their characterization, we have used scanning electron microscope, UV visible spectroscopy, FKR, and X-ray diffraction. And for biological studies, the animals, biopsy mice were divided into 6 groups and each group having the 6 mice. Group 1 is the normal control group in which the mice were without any problem and healthy. Group 2, the mice, this is the negative control and monosodium urate crystals have been induced in them. Group 3, allopurinol positive control in which the commercially available treatment has been provided. Group 4, 5 and 6 are the treatment groups in which the curcumin nanoparticles with a concentration of 5 ppm, 10 ppm and 20 ppm have been administered to the mice. So this scheme shows the details of these treatments. Total 6 groups. First week is only for their normal settlement and the second week group 2 to 6. Group 2 to 6 they administered with the treatments and induction of hyperuricemia by monosodium urate injection in the ankle joint, and in the third week this induction have been done in the peritoneal cavity. Then fourth and fifth week is a treatment of hyperuricemic mice with 5, 10 or 20 ppm of curcumin nanoparticles, and allopurinol and nanoparticles have been administered orally. Then animals were sacrificed at the start of the 6th week. After the completion of study, the biochemical analysis of these animals have been also performed in the liver function test, renal function test, inflammatory markers, lipid profile, and xanthine oxidase levels have been analyzed. These are the results obtained with the UV visible, in which the peak at 360 to 340 to 360 confirmed the presence of synthesized curcumin nanoparticles. And FKR spectrum of curcumin nanoparticles showed that the intense band of OHCH, suitable bond C, and carbon-oxygen groups are present in the curcumin nanoparticles. Some analyses have also shown that the well-defined crystalline structures of these spherical-shaped nanoparticles of curcumin nanoparticles have been observed with a mean size of 21 nanometer. XRD also showed the good results, and it gives human nanoparticles was found at 20 to 1810, which corresponds to the energy space. For the safety evaluation, the renal function test and kidney function test have been performed to check out the effect of human nanoparticles on the kidneys and liver. Here we can see that the monosodium urate crystals badly affect the kidney, like uric acid level 10 milligram. Here it is urea 53 milligram and 1.15 creatinine level. Whereas by using the curcumin nanoparticles with the 5, 10 and 20 ppm, we can see here the significant difference or decline in the levels of uric acid as compared to the control group with the negative control. So here we can see that the 20 ppm curcumin nanoparticles provided a very good result, and in the decline of the uric acid concentrations 1.95 and urea 1.20 and creatinine level have been controlled under 0.25 milligram per deciliter. And in the liver function test, same thing we have observed that the 5 ppm, 10 ppm and 20 ppm carbon curcumin nanoparticles provided a good result as compared to the negative control and allopurinol, in which the 20 ppm showed best results to control the ALT, AST, total bilirubin and ALP levels of the liver. Some serum inflammatory markers have been also identified, in which serum interleukin 10, tumor necrotic factor alpha, serum interleukin 6 and serum interleukin 1 beta have been observed. And in all these graphs, we can clearly found that the all the inflammatory markers shows a very good result, in which the 5 ppm, 10 ppm and 20 ppm treatment of curcumin nanoparticle controlled or showed the inflammation level in all these markers. Here this is the xanthine oxidase level, which showed the best result with 20 ppm, although the 10 ppm also showed very good result, and here the xanthine oxidase level in serum both are significant level with 10 ppm and 20 ppm. Serum lipid profile have been also observed, in which the serum cholesterol level have been decreased as compared to the control group by using the 20 ppm curcumin nanoparticles, and serum triglycerides are also controlled. Serum LDL showed a good result with 10 ppm, 20 ppm both, and serum HDL level also showed a good result with the 10 and 20 ppm. And with 5 ppm also, but there is a little bit significant difference with the 20 ppm. So by concluding our study, we can say that the curcumin nanoparticles exhibited significant ameliorative effects by successfully reducing xanthine oxidase activity, serum uric acid levels, and inflammatory markers. So these findings suggest that the curcumin nanoparticles may serve as a promising alternative to conventional therapeutic approaches. These are the references, and thank you for your attention. If you have any question, you may ask.
Thank you so much, Dr. Razia, for sharing your beautiful insights with us. Now let's welcome our fourth presenter, and he is from University of Agriculture, Faisalabad, Dr. Rai Muhammad Nasir. Dr. Rai Muhammad Nasir would be presenting today advanced RNA-based personalized cancer vaccines, neoantigen targeting and delivery innovations. Welcome, Dr. Saab.
Assalamu alaikum and hello to everyone. I am Dr. Muhammad Nasir, Assistant Professor in the Department of Pathology, University of Agriculture, Faisalabad. My topic of presentation is advances in RNA-based personalized cancer vaccine and also neoantigen targeting and delivery innovations. The background of our study is that the limitations for the treatment of the cancer, because the cancer have the property of tumor heterogeneity, also have the property of immune evasion, and there are many reported side effects of chemotherapy or immune therapy for the treatment of the cancer. The RNA-based vaccines designed to stimulate tumor-specific neoantigens. These RNA-based vaccines represent a new frontier in immunization, offering a faster and more effective way to combat infectious diseases, as demonstrated by the rapid development of messenger RNA vaccines in the recent disease that was reported, and that was COVID-19. These vaccines leverage new technologies for delivery and stability, expanding their potential beyond COVID-19 to other viral diseases and revolutionizing public health. There are also some property of inducing effective cytotoxic T-cell responses, and our study was inspired by the successes of messenger RNA vaccines for the treatment of infectious diseases. That's why we have been studying the messenger RNA vaccines for the treatment of the tumor. How messenger RNA vaccines work? Messenger RNA technology used in this study or in this work, like messenger RNA vaccines deliver genetic instructions to the body cells, and these instructions direct the cell to produce a specific protein such as the spike protein from the virus. And then immune system recognizes these foreign proteins and mounts a defense, preparing the body to fight for the actual virus. The aim of our study is exploring innovations in neoantigen identification, vaccine design, RNA delivery system. Neoantigen identification is the identification of proteins that produce from the mutation of the tumor, and then our immune system recognizes these mutated proteins, and we can try to use this technology, this mutation proteins, for the treatment of the tumors. The methods which we used include the systematic review of the recent studies that involved from 2022 to 2025, which focuses on the neoantigen prediction strategies, RNA engineering method, delivery platforms of different carriers, polymeric carriers or exosomes, and also clinical trials outcomes involved in our method. The innovations in the neoantigen identification are next generation sequencing, advanced computational prediction tools that enables highly personalized vaccines. Lipid nanoparticles are highly efficient and also have a great stability. Polymeric carriers also have biocompatibility and flexibility. Exosome-based delivery has a natural cell-derived carrier, cell-amplifying RNA, lipoplexes, enhanced expression and durability. The clinical advancement which have been reported that are the combination of messenger RNA and pembrolizumab that reduces the reoccurrence of the tumor, that is melanoma. There is also autogene cevumeran improves the survival in pancreatic cancer, and this is an experimental messenger RNA based individualized cancer vaccine that is developed by two companies that are BioNTech and Genentech to help the immune system target to attack a patient's own cancer cell, preventing reoccurrence after surgery. It is made from a patient's tumor cells, and the messenger RNA vaccine is designed to train the immune system to recognize and destroy cancer cells by encoding up to 20 personalized tumor neoantigens. It is being studied in clinical trials for various cancers, including pancreatic cancer, to enhance anti-cancer immune responses. There are also many evidence of these kinds of combination and also therapies. The key outcomes in our studies, which we got from different reviews, are the enhanced delivery efficiency, improved antigen presentation, reduced dose requirements, longer lasting immune responses. The future perspective of these studies could be the integration with checkpoint inhibitors, expansion to multiple cancer types, potential to redefine standard cancer therapies in the future. The conclusion of our study is that RNA-based personalized cancer vaccine represent a turning point for the treatment of cancers, because cancers have a different kind of mutations at the different exon level. and these kind of RNA-based vaccine can make proteins that our immune system can detect and can be used in the treatment of the cancer. The neoantigen therapy also have been used, like we can say the transformative immunotherapy, that is a future which uses a patient's unique tumor mutations as a precise cancer vaccine target, which advanced delivery system enhances the immune response to cancer, leading to more effective and personalized immunotherapy by identifying and delivering these tumor-specific neoantigens with innovative technologies like nanoparticles. Researchers aim to generate powerful, durable, and tumor-specific T-cell responses, ultimately providing tailored treatments that eliminate cancer cells and prevent the reoccurrence of the tumor. The ongoing clinical trials show promising outcomes, and these kind of combinative therapies can deliver good treatment for the cancers and for the reoccurrence of the cancers. So these are the reference we used in our study that was taken from the different studies of reviews from 2020 to 2025. Thank you so much. If you have any question, you can contact with me. Like we also share our email. Thank you.
Thank you so much, Dr. Rai Muhammad Nasir, for joining us. Next, we have Vanhar Afzal from Punjab University, and he would be presenting today toxicological evaluation of cardiac damage by bisphenol S and polystyrene nanoplastics co-exposure in rats.
Assalamu alaikum. My name is Vanhar Afzal, and I am an MPhil zoology scholar from the Department of Zoology at the University of the Punjab, Lahore. The topic of my presentation is toxicological evaluation of cardiac damage by bisphenol S and polystyrene nanoplastics by the co-exposure in rats. Cardiovascular disease is a collective term for a spectrum of pathological conditions that adversely affect the heart and vascular system. The World Heart Federation shows a severe 69% rise in CVD-related deaths. Between 1990, it was 12.1 million, and in 2021, it has become about 20.5 million. CVDs are largely driven by both modifiable and non-modifiable risk factors, with the modifiable risk factors accounting for about 75% of the CVD cases. Recent findings from the Global Burden of Disease study 2019 indicate that environmental pollution played a role in nearly 62% of all cardiovascular related deaths worldwide. Among these environmental pollution, plastic pollution has recently surfaced as a novel and growing concern, linked with about 46 to 50% of the CVD cases. Among all types of plastics, polystyrene has emerged as a key industrial plastic, representing about 5.4% of the Europe plastic manufacturing output. Now, when exposed to the environmental conditions, these plastics products gradually degrade, first breaking into the microplastics and ultimately fragmenting into the even smaller nanoplastics, which have the capability to easily cross the physiological barriers. Plastic formulation regularly utilizes chemical adjuvants at the rate of about 9.75 million tons by 2024. Among the chemical adjuvants, bisphenol has emerged as the primary substitute in the consumer products. Recently, it was found that BPS exposure activates harmful mechanisms in the heart, including myocardial remodeling, thickened heart walls, collagen buildup, and inflammation. An alarming fact is that PSNP can strongly bind to the hazardous substances like industrial chemicals, toxic metals, and plastic additives, increasing their biological health risks. A 10-year population study highlighted that prolonged exposure to the plastic plus the additives is linked to the 46 to 49% higher risk of CVDs. Now, if I talk about the objective of my research, the first objective of my research is to evaluate synergistic severeness. The second one is to evaluate the abnormalities by the synergistic severeness as well as alone, plus environmental toxicological knowledge and informing plastic related safety regulations. The methodology which I have used in my research include 24 male visitor rats with the age of about 8 to 10 weeks and weight of about 180 to 200 grams. I have divided the 24 male visitor rats into 4 groups with 6 rats in each group. The first group is control group with no exposure. The second group is BPS group with the 1 ppm exposure of the PPS. The third group is PSNP group with the exposure of 1 ppm concentration of PSNP. And the fourth group is co-exposed group with the exposure of mixture of BPS plus PSNP at the concentration of 1 ppm. All of the solution was prepared in the filtered water and were given by the glass bottles with the stainless steel sippers. For the sampling, I have used anesthesia. After the 28th day of the administration and acclimatization and this exposure, I have humanely euthanized the rats by the use of anesthesia, ketamine xylazine mixture at the concentration of 50 and 6.8 milligram per kg. For the sample collection, I have used the whole blood by the cardiac puncture and stored the extracted organs at minus 80 degrees Celsius. Now, if I talk about the sample processing, I have used the biochemical analysis in which I have analyzed two types of parameters. The first is the parameter for the dyslipidemia, which include triglyceride, HDL, LDL, and total cholesterol. And the second one is for the cardiac enzymes, which include CKMB and LDH. For the histopathological analysis, I have used a detailed protocol and the microscope with 10x lens objectivity. The results which I have taken from the research include, first of all, the results for the dyslipidemia parameters. Triglyceride. One way ANOVA revealed significant hypertriglyceridemia in the co-exposed group, while the individual exposure showed no significant change. If I talk about the total cholesterol, significant rise has been observed in both PSNP and PSNP plus PPS. However, serum HDL-C levels showed significant decline across the study group. Statistical comparison showed significant HDL-C reduction in both PSNP and mixture PSNP plus BPS group. If I talk about the LDL, post-hoc analysis confirmed that none of the treatment group demonstrated significant difference. Now, the second type of parameters which I have evolved include the cardiac enzymes. Post-hoc analysis revealed that only PSNP plus BPS exposure group showed statistical higher LDH activity, while neither PSNP nor BPS alone produced significant changes in the biomarker. CKMB. Post-hoc analysis demonstrated that both PSNP and PSNP plus PPS group showed markedly increased CKMB concentration relative to the controls. If I talk about the histopathological analysis, the abnormality which I have analyzed in my research are interstitial hyperplasia by the brown arrows, tissue rupture by the red arrows, disrupted myofiber integrity by the black arrows, and vascular congestion by the pink arrows. PSNP plus BPS group exhibited the most severe histopathological changes indicating potential additive cardiotoxic effect. If I talk about the study gaps which my research has fulfilled, the first study gap is synergistic cardiac injury, severe cardiotoxicity in a synergistic manner. The second is a proposed mechanism from lipid dysregulation to the structural damage. Third one is linking structure to the function. The pileup of the lipid molecules in the cardiac muscles cause the structure rupture, which lead to the dysfunction of a number of cells. The fourth and very important point which I have observed in my study is the Trojan horse. Nanoplastic act as a Trojan horse for the BPS, potentially enhancing its disruption and biological uptake. The broader implication is that the magnification of the toxicity upon combination is a novel and critical insight. It suggests that the cardiovascular disease from common environmental contaminants like BPS may be significantly underestimated when not considered in the context of co exposure with other pollutants like nanoplastics. As per the conclusion is co-exposure to BPS and PSNP induces synergistic cardiotoxicity. The dyslipidemia leads to the structural heart damage, resulting in functional myocardial injury. Implications and future directions. We should consider the cocktail effect of the co-exposure to common environmental pollutants like plasticizers and nanoplastics in cardiovascular risk assessment. Risk assessment framework should evolve from the single chemical evaluation to mixture-based toxicity testing. Further investigations are warranted to elucidate the precise mechanisms by which this combination disrupts lipid metabolism and mitigate this damage. Lastly, I want to acknowledge the ultimate efforts made by my supervisor, Dr. Nabila Rohi, my senior lab fellows, Dr. Kaleem Maksood, and Ms. Farwa Liaquat, and I want to express my sheer and sincere gratitude to my dedicated research fellows. Thank you.
Next, we have Iman Murtaza, and she's also from Government College University, Faisalabad. She would be presenting today ameliorating nickel toxicity in silver carp with coriander seed extract.
Bismillahirrahmanirrahim. Assalamu alaikum. My name is Iman Mutsa. I am MPhil student from Government College University, Faisalabad. First of all, thank you for giving me opportunity in this e-presentation of 8th International Conference on Applied Zoology 2025. My presentation topic is ameliorating nickel toxicity in silver carp. Their scientific name is hypophthalmic thismolytics with coriander seed extract. Here's the introduction. Heavy metal pollution is a serious issue for aquatic ecosystems because it results in a variety of toxicities that have a significant impact on aquatic faunal populations. Nickel is especially toxic, affecting fish metabolism, growth organs, and behavior. Grass carp, a major aquaculture species, is vulnerable to heavy metal contamination. Chelating agents help detoxify heavy metals. Natural options like coriander show strong potential. Coriander seeds contain antioxidants and metal binding compounds are inexpensive and widely available. I researched on silver carp because it has a filter feeder, have low price and fast growth rate, also plentivores and a superb flavor. That's the material and methods. First, the fingerlings of hypophthalmic thismolytics purchased from a hatchery and acclimated for 2 weeks under optimum conditions like temperature and pH. After that, nickel chloride solution prepared with deionized water. Then preparation of coriander seed extract. After the preparation of coriander seed extract, the formation of feeding pellets is done. After all the procedure, the statistical analysis is done. These all are operators that are used in this research for different purposes like growth estimation, crude protein, herbal extract, ash, and gross energy. Here's the ingredients composition of experimental diets and the results of the growth performance of hypophthalmic thismolytics of 60 days feeding with coriander seed extract along with exposure to sublethal levels of nickel toxicity show optimum results at 1%. At the same case of carcass, it also showed best results at 1%. and hematological parameters of hypothalamic thismolytics after 60 days feeding with coriander seed extract along with exposure to sublethal levels of nickel toxicity show best results at 1%. So the conclusion of the research is the result of the current study demonstrated that CC, especially when giving at a dose of 1%, may lessen nickel toxicity in hypophthalmic thismolytics. Nickel exposure caused a significant slowdown in blood parameters, carcass quality, growth, and antioxidant enzyme activities, and has negative effects on biochemical indices. Coriander's chelating abilities allowed to mitigate the negative effects of nickel, which is improved the growth, carcass, hematology, and antioxidant status of hypophthalmic thismolytics. Thank you for your attention.
Thank you so much, Iman Murtaza, for joining us. I would like to welcome Sayyid Muhammad Farhan Ali Shah from Government College University, Faisalabad, and the title of his talk is Black Pepper Seed Extract Mitigates Nickel Toxicity in Grass Carp.
Assalamu alaikum. This is Sayyid Muhammad Farhan Ali Shah, and my topic of research is black pepper seed extract mitigates nickel toxicity in grass carp, Pterophragmodon idella. As you know that health of aquatic species is negatively impacted by heavy metal contamination. They can alter organisms' physiology and biochemistry. In all living organisms, nickel is necessary trace metal. Both natural and human-caused elevated nickel levels in aquatic environment can have harmful toxic effect on aquatic organisms. Grass carp is highly prized freshwater fish that is widely farmed across the globe, mostly in Asia and Europe. Herbal therapies has numerous advantages for fish. Black pepper contains piperine as the main bioactive component. Numerous investigations have proven piperine's capacity to squelch radicals. The aim of this research was to investigate the mitigation potential of black pepper seed extract supplementation against nickel stress in C. idella. The objectives of this research are, first of all, improving growth performance, body composition, hematology, and histology. Secondly, the objective of this research was mitigation of negative impacts of nickel on growth performance, body composition, hematology, and histology. Methodology. First of all, fingerlings were acclimatized in V-shaped tanks for 2 weeks, and stock solution was prepared by mixing nickel chloride with deionized water. For black pepper seed extract preparation, black pepper seeds were ground into a fine powder and the herbal extract was obtained using a Soxley device. Then feeding pellets were formed by grinding the feeding material to pass through a 0.5 mm sieve, mixing components and adding fish oil and black pepper extract. There were 6 experimental diets were created using cottonseed meal as the basal diet. Number 1, the negative control group was fed basal diet without nickel toxicity, while the positive control group was exposed to nickel toxicity. The remaining 4 groups faced 3.6 mg per liter nickel toxicity and were supplemented with 1%, 2%, 3%, and 4% black pepper seed extract. Each tank's fish were bulk weighted at the end of the trial to assess the growth. The fingerlings' growth performance was assessed using standard formulas. Body composition of fingerlings was determined by using standard protocols. Jaldal apparatus was used to evaluate the crude protein content. Crude fat was estimated using Soxley apparatus. Moisture content was determined by oven drying at 1.105 degrees centigrade for 12 hours. Ash was determined by burning in electric furnace. For hematological studies, white blood cells and red blood cells are counted using Newborn counting chamber and hemocytometer. Standard formulas were used to calculate the indices of red blood cells like mean corpuscular volume, mean corpuscular hemoglobin, and mean corpuscular hemoglobin concentration. At the end of experimental trial, fish were anesthetized using clove oil. Tissue samples were fixed in 10% formalin, then stained with metaxylene and eosine stain. After drying the tissues with 96% ethanol, they were embedded, sectioned, mounted, and stained for histological examination. Results: growth performance. Highest final weight and weight gain was observed at negative control. Second highest final weight and weight gain was observed at 1% supplementation level. Lowest final weight and weight gain was observed at positive control. Similar results were observed with body composition and hematology. Histological study results shows negative control group showing normal central vein and normal hepatocytes. Positive control showed necrosis with highest level of nickel accumulation and contracted central vein, showing the 1% group shows normal central vein, while 2% showing some amount of nickel accumulation, 3% shows highly degenerated hepatocytes along with a dilated central vein, while 4% also showed highly degenerated hepatocytes, dilated central vein, along with some extent of nickel accumulation. Conclusions: it is concluded that addition of black pepper seed extract mitigated nickel negative effects of nickel toxicity on growth performance, hematology, body composition, and histopathology. Thank you.
Thank you for joining us. Next, we have Sana Amjad from US. She would be presenting today evaluation of the anti-staphylococcal potential of honeys from diverse origins. Let's welcome Sana Amjad.
Assalamu alaikum, respected chair, distinguished scientists and fellow delegates. I am Sana Amjad from University of Veterinary and Animal Sciences, and it is an honor to present my research title, Evaluation of the Anti-Staphylococcal Potential of Honeys from Diverse Origin. Honey is not just a natural sweetener, it is one of the oldest therapeutic substances known to mankind. Historical records, including prophetic and Greek medicine, describe honey as a healer. Modern science confirms that honey possesses antibacterial, anti-inflammatory, and antioxidant, and even wound healing properties. Globally, over 1.2 million tons of honey are produced worldwide each year, with China, Turkey, Argentina, and US are the top producer of honey. The antibacterial properties of honey are because of low water content, high viscosity, acidity, hydrogen peroxide content, and non-peroxide component, particularly methyl glyoxal, all contribute to these properties. One of the earliest traditional medicine still in use today for treating variety of human bacterial infections and can reduce microbial pathogenicity. As well as Although honey is widely known for antibacterial properties and its effectiveness varies and depending on floral source, geographical origin, and processing method. On the other hand, genus Staphylococcus bacteria with more than 30 species and 30 species are responsible for skin infection, food poisoning, wound complications, and antibiotic resistance. So, the central question of my research was, can natural honey serve as an affordable, accessible antibacterial agent against Staphylococcus infections, and which type of honey works best? And statement of the problem is that Staphylococcus is represented by more than 30 species, and many of them are pathogenic. Honey has natural antimicrobial potential and it could be effective natural antibacterial agents. That's why it is essential to study honey inhibitory effect against the Staphylococcus species. The main objective of my research was to assess the bactericidal potential of different types of honey on Staphylococcus species, as well as to identify which honey source are most effective. And hypothesis. Bactericidal potency of honey varies with the type of honey. If we talk about methodology, this research was conducted in Department of Wildlife and Ecology, US Lahore, and the samples are also collected from Department of Wildlife and Ecology, US Lahore, Pakistan. Bacterial strains used Staphylococcus aureus, Staphylococcus hemolyticus, Staphylococcus hominis, and Staphylococcus intermediate. And we use the agar-well diffusion method to assess the antimicrobial properties of honey. Basically, honey samples were honey solutions were prepared at different concentration: 25% concentration, 50% concentration, and 75% concentration. Agar well diffusion methods was used, and wells are made, and in which different concentration of honey are poured and incubated at 37 degrees Celsius for 24 hours. And at the end, zone of inhibitions were recorded to measure the bacterial growth. Degrees Celsius, 50 degrees Celsius, and 75 degrees Celsius were added to these wells using a micropipette. The plates were then incubated at 37 degrees Celsius for 24 hours. Mean Diameter of inhibition zones were recorded in mm. All the trials were completed in triplicates for each of honey concentration, and data analyzed through ANOVA using SAS9A. The result of my research was all types of honey shows antibacterial property, but the level varies significantly. The most sensitive bacterium was Staphylococcus aureus, Hemolyticus, Staphylococcus intermedius, and Staphylococcus huminus. This figure shows the zone of inhibition of different types of honey against Staphylococcus species. And this is the table with mean zone of inhibition of wild honey against Staphylococcus aureus. T1 represents Staphylococcus aureus, T2 Staphylococcus hemolyticus, T3 Staphylococcus intermedius, and T4 Staphylococcus hominis. And this is the table showing the zone of inhibition of formed honey against different type of Staphylococcus. And this is the mean zone of inhibition of marketed honey against different type of Staphylococcus aureus. When compared the effect of different type of honey against Staphylococcus species, then the result was observed that Staphylococcus aureus was more sensitive than other species of Staphylococcus. When we compared the type of honey, wild honey demonstrated the high antibacterial potency. When comparing the farmed and marketed honey, farmed honey consistently showed larger inhibition zone, and when we compare the marketed honey and farmed honey as unmarked, in which um, from marketed honey, Aslul Jabal showing higher zone of inhibition as compared to other Karshi, Forever, Young Honeys, and Marhaba. The expected outcomes was the finding of the present research would help the consumers to understand the antimicrobial effect of different types of honey against various Staphylococcus infections. Honey, at the end, I will include my research. Honey is not just food, it is a natural antibiotics. Wild honey shows the strongest antibacterial effect, especially against Staphylococcus aureus. Farmed honey outperform commercial marketed honey, including that processing may reduce potency. In the future, honey could be developed as complementary alternative to synthetic antibiotics, especially in the era of drug resistance. Thank you for your patience and attention. I welcome questions, feedbacks, and collaborations.
Next, we have Iram Rashid from Government College University, Faisalabad, and the title of our talk is New Insights into the Effect of Polylactic Acid Microplastics on Performance of Labioheta Fingerlings.
Assalamu alaikum. I am Dr. Iram Rashid from Department of Zoology, Government College, Faisalabad. Here is my topic, a new insight into the effect of polylactic acid microplastic on the performance of Labioheta fingerlings. Here is my contents of my presentation. Here is an introduction. Aquaculture is the fastest growing fruit production industry around the globe. Fish is a good source of protein for humans, accounting for 16% of animal protein ingested by the world population. Fish meal is a good source of protein content and essential amino acid profile has long been the primary source of protein in aquafeed industry. There is a two type of plant that I had used during my research work, sunflower meal and canola meal. Number one is sunflower meal is a good source of protein and plant oil. Sunflower is a low cost, easily obtainable plant source of protein that is both palatable and nutritious for fish. And microplastic. Actually, microplastic refers to the plastic particles having a diameter smaller than 5 millimeter. According to statistics, aquatic environment get roughly 14 million tons of plastic trash each year. There is a two type plastic, non-biodegradable microplastic and biodegradable microplastic. Here are some effects of microplastic. Being aquatic pollutant, microplastic can accumulate into the body of aquatic animals, having detrimental physiological and physical impacts on them. Previous studies research have confirmed the negative impact of microplastic on fish growth, feed intake, survival, metabolism, immunity, and other toxic responses. The most important conducive for microplastic from land environment to the freshwater environment are the Liburihita. Liburihita is a column feeder, highly palatable and consumer choice. Here is some sources of microplastic. This diagram shows how microplastic enters into the body of the living organism, especially into the body of the fish. Here is some sources, industries, domestic activities, and coastal activities. All of these participate for the accumulation of microplastic into the body of a fish. These coastal activities also play important role for the dispersal of the microplastic into the body of the fish. And other domestic activities and industrial activities also play important role for the microplastic dispersal. Now here is some contents of this presentation. I've to determine the impact of microplastic supplemented oilseed meal based diet on growth performance of major carps. To evaluate the impact of microplastic supplemented seed meal based diet on body composition of the major carps. To investigate the impacts of microplastic supplemented oilseed meal based diet on nutrient digestibility of major carps. To evaluate the hematology and mineral analysis of the major carbs. Now, materials and method. The fingerlings were brought from the government fish hatcheries located on the Satyana Road, Faisalabad. The fingerlings were brought into the fish nutrition laboratory for the 2 weeks in fish shaped tanks. After that, 5% saline solution was used to get rid any ectoparasite and prevent from further parasitic infections. And the aeration was used throughout the capillary system. Here's the water quality parameter that I used during my research work: pH, temperature, and dissolved oxygen. All of these parameters were maintained throughout the trial period. Feed ingredients and experimental diet feed. Commercial feed ingredient analyzed for the chemical composition. Test at 1 contains 0% microplastic, means no microplastic control. It is also controlled. Test type contains 0.5% microplastic. Test type 3 contain 1% of microplastic, while test type 4 contain 1.5% of microplastic. Test type 5 contain 2% of microplastic. Test type 6 contain 2.5% of microplastic. Here is the ingredient, all composition of these research work. The formulation of pellets, the feed ingredient is finely grinded to pass through 0.5 millimeter sieve size. All the ingredients mixed in the mixture for 5 minutes and the fish oil gradually added. After that, 10 to 15% water was added to prepare the suitable dough. After that, it was processed by pelleting machine to make a feed pellets. This is the feeding protocol and sample collections. Uh, Triplicate tanks, 15 fingerlings was used. In triplicate tanks, feeding rate was the 5% of the live weight body weight. After the 2 hours of feeding session, the tanks were washed and the pieces were collected. After that, the pieces were dried in the oven and stored for the chemical analysis. Here is the formulae to determine the hematological indices and growth digestibility performance and digestibility calculation, weight gain percentage, SGR and FCR. The chemical analysis of feed, feces, and body composition. The crude protein was determined with the help of microgel dal operators, and crude fat was determined with the help of SOX operators. Ash was determined with the help of electric furnace, and the gross energy was determined with the help of oxygen bomb calorimeter. Statistical analysis, rate of growth, nutrient digestibility, carcass composition, hematological indices, and absorption of fish were determined with the help of one-way ANOVA, means comparability honesty test, and the co-state computer package was used. Now, growth performance. The growth performance of table 2 shows the growth performance of labiorhita fingerlings fed from biodegradable polylactic acid microplastic. Significant differences indicated by unique superscript with rows. Test type in this column, test type are shown 1 to 6. Polylactic acid microplastic 0 level to 2.5% microplastic. Initial weight was 0.07, but the final weight was 0.2, and the weight gain percentage, the test type 1 contained 15.97 weight gain, while type 6 contained 6.13 weight gain. As the microplastic concentration increased, the weight gain was also increased. Similarly, as the microplastic concentration increased, but the feed intake decreased. And the specific SGR, as the microplastic concentration increased, the SGR was decreased. Here is the apparent nutrient digestibility. Table 3 shows the apparent nutrient digestibility of labiorhita fingerlings fed polylactic acid microplastic, significant indicated by unique superscript with the rows denoted with statistical variance. Test type 1 contain analysis of feed. This shows the analysis of feed. This shows the analysis of feces. In test type 1, crude protein, crude fat, and gross energy. The crude protein was in 9.14 in test type 1, while the crude protein 15.27 was noted in test type 6 after the 2.5% of microplastic dose. Test type 6 crude fat in test type 6 63.42 was noted as compared to the 7 compared to the test type 1. As the microplastic concentration increases, the apparent nutrient digestibility was decreased. With the increase of microplastic concentration, the apparent nutrient digestibility was decreased. But in case of analysis of feces, with the increase of the value of the crude protein, crude fat, and gross energy was also increased. Here is a table 4. Shows a hematological analysis of the labiorhita fingerlings fed biodegradable polylactic acid microplastic with sunflower meal based diet. Test type 1 shows the PCV, MCV. Test type 1 shows at 26 point in case of MCV. The highest value of the MC was seen in test type 6 as compared to the test type 1, say 161.73. Mineral analysis table 5 shows a mineral analysis of labial plastic. It shows the calcium, copper, chromium, potassium, tin, manganese, selenium, nickel, sulfur, phosphorus, manganese, selenium, nickel, sulfur, phosphorus, and sodium. The highest value of the that contains 0% microplastic, which is 156.41. The lowest value of the calcium was seen in test at 6 to 150.35. This is a similar in other chromium, copper, ferric, and etc. The highest value of the zinc was diet. The highest value was seen in test diet 1 6.02, and the lowest value of zinc was seen in test diet 6 test diet, and the lowest value of zinc was seen in test diet 6 test diet 6 2.5% of microplastic, which was 5.51. Here is the figure this that shows the crude protein, crude fat, ash, and moisture of labio heat of fingerlings, fat, sunflower, polylactic acid, microplastic. Now here is a conclusion. This research studies confirm that biodegradable microplastics per year in a level-dependent manner resulted in reduced growth, performance, feed utilization, and nutrient digestibility in labio heat of fingerlings. Microplastics sphere also significantly altered the carcass composition of fingerlings by increasing their body fat and decreasing their protein content. Blood parameters of Labiorhita fingerlings also affected remarkable by biodegradable microplastics per year. Resulted showed that higher level of biodegradable microplastics resulted more damage to the health of fish as compared to the lower level. Now, thank you.
Thank you so much, Iram Rashid, for joining us. Now let's moving forward. Our new presenter is Dr. Sana Farouk from ComSets University Sahiwal campus. She is presenting today her work, which is entitled as Seromonitoring and Molecular Detection of Newcastle Disease Virus in Non-Vaccinated Birds of Pakistan.
Assalamu alaikum dear participants. My name is Sana Farouk and I am from ComSets University Islamabad Sahiwal campus. And today I am here to represent the topic of seromonitoring and molecular detection of Newcastle disease virus in non-vaccinated birds of Pakistan. In this presentation, we will go through introduction, research methodology, results, and conclusions. Newcastle disease is a common respiratory disease caused by Newcastle disease virus, which belong to family Paramyxoviridae and genus Aviola viruses. We can classify Newcastle disease virus in 3 different pathotypes according to their severity. Lentogenic is less severe, show less signs and symptoms, and usually used as live vaccine. Pathogenic show intermediate mortality and pathogenicity. It usually show digestive and respiratory signs, while virogenic is highly pathogenic and it show neurological and respiratory signs and have high mortality and morbidity rate. Newcastle disease can cause huge economic losses as our GDP has as 1.3% of our GDP depend upon the poultry production. So it can cause a huge loss. A single disease can cause a huge loss in our economy. Symptoms of Newcastle disease virus. Newcastle disease respiratory, digestive, and neurological symptoms. The simple clinical cases are characterized by sneezing, respiratory distress, green diarrhea, while the neurological symptoms are curling of neck, circling, weakness, tremors, and paralysis in extreme cases. Statement of problem. As we know that vaccination is a common practice among commercial poultry to save broilers, layers, and breeder folks from different kind of infection and outbreaks. But still, poultry industry is facing different disease outbreaks in Pakistan. So it is suspected that non-vaccinated poultry is behind the onset of these infections because they are reared in natural environment and the vaccination is completely eliminated in these areas or it partly practiced. Therefore, this study is conducted to confirm the status of non-vaccinated birds. Aims and objectives. The aim of our current study was the serological and molecular assessment of NDV in non-vaccinated birds of Pakistan. These aims can only be achieved by following objectives: to elucidate the serological status of NDV in non-vaccinated birds of Pakistan, then the NDV isolation from non-vaccinated birds of Pakistan, and the description of molecular status of NDV in non-vaccinated birds of Pakistan. Methodology. This specific study is carried out during 2000 reference lab for poultry diseases. It has its regional collection centers throughout Pakistan, which include Punjab, KPK, Sindh, Islamabad, through which we receive enzyme-linked immunoglobulin assay, which is carried out through IDEX kit following their manual. And for virus isolation, we use in novo inoculation technique and cloacal and tracheal swabs and tissue samples were processed. Detection RNA extraction is carried out through Kaizen kit and polymerase chain reaction is carried out by in vitrogen. Results: the maximum number of from Punjab followed by ICT, KPK and Sindh as shown in the figure. Three types of samples were collected: sera samples, cloacal and tracheal swabs, and organ samples. Non-vaccinated birds, species investigated for ND. Different type of birds which are investigated for Newcastle disease virus, which include wild domestic, wild migratory, and backyard poultry. And different numbers of birds are mentioned, among them naked neck has 94 samples, which is the maximum number. Serological evaluation. Out of 344 samples which were subjected to hemagglutination inhibition assay, Punjab show the maximum HI log 2 titer. Then ICT followed by subjected to the test. Among those 207 samples, only 10 show positivity, which show 4.8% of viral isolation. And the figure show us the viral isolation is maximum in summers, and the molecular reduction and viral isolation presents that NDV can be harbored and transmitted through non-vaccinated poultry to commercial farms during interregional trade and movements. I conclude this study with the fact that non-vaccinated birds can serve as a reservoir for NDV and can cause a huge economic loss in Haiti.
Thank you so much, Dr. Sana Farouk, for sharing your insights with us. Let's welcome Dr. Sabah Farouk. And she's from Allama Iqbal Open University, and she would be presenting today development of post-vaccination baseline against M. Gallus septicum in vaccinated birds and efficacy assessment of commercially used vaccines. Let's welcome Dr. Sabah Farouk.
Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Sabah Farooq from Allama Iqbal Open University, and I'm really privileged to be a part of 8th International Conference on Applied Zoology 2025. Today I will be talking about development of post-vaccination baseline against Mycoplasma gallosepticum in vaccinated birds And the efficacy assessment of commercially used vaccine in poultry. Mycoplasma galisepticum is a etiological agent of chronic respiratory disease, and this disease is responsible for huge economical losses. Uh, the disease may be complicated by certain bacterial and viral pathogens, which include Newcastle disease virus, infectious bronchitis virus, and bacterial pathogen includes E. coli. Now, even in the absence of these secondary complicating infectious agents, the disease may be misdiagnosed or undiagnosed due to its asymptomatic nature. But for farmers to control this disease, there are two uh, strategies. First one is preventive strategy by using vaccines, and second one is therapeutic strategy. When disease is expressed in the flock, it is managed by use of certain antibiotics. One of the most important challenge caused by Mycoplasma galisepticum infections is due to its vertical transmission from infected parental flock to the offspring. This vertical transmission may result in economical losses due to reduced hatchability, reduced egg production, and low meat quality in all kinds of commercial birds, including broiler, breeder broilers, and layer birds. Now, the infection may be controlled by different strategies, and flock free of infections are of great importance for farmers. So, to control these infections from vertical transmission, with use of vaccine has been in practice, but still reports of infection has been very common. So the present study was designed to evaluate the efficacy of commercially used vaccines against Mycoplasma galisepticum infections and compare it to the exposure titer of vaccinated birds. For this purpose, the study was divided into two clusters. First included the development of post-vaccination baseline by commercially available live vaccine and kill vaccines. Live vaccine which was used in the study is FVAX MG by Intervet, and kill vaccine which was used in the present study was MG-BAG by Zoetis. Both vaccines were administered at the designated age to 30 birds, experimental birds, which were kept in separate chicken isolators. A control group of 10 birds was also placed in a separate chicken isolator. Their cross-contamination was controlled by strict biosafety and biosecurity measures. After 2 weeks of interval, these birds were bled and serum was collected, which was then subjected to indirect ELISA. Now, to compare this baseline with the exposure titer, a longitudinal study was conducted uh, in commercial farms. These farms were three different flocks were selected, and these flocks were of almost 1 to 2 years. Samples from these selected flocks were studied by indirect ELISA and were subjected to molecular detection by PCR. During the study, same conditions were maintained for both experimental group and control group. To assess the efficacy of killed vaccine, three different flocks were studied. Two were from the same farm and one farm was different. The study time period was from February 2017 to March 2019, February 2017 to October 2017, from 14 week to 47 week, from May 2017 to March 2018, and it was of 9 to 44 week of age, and the last flock was observed from June 2018 to March 2019, and this flock was of 7 to 51 weeks of age. Results of vaccination antibody titer evaluation by ELISA gave us understanding that the maximum antibody titer was achieved with high antibody titer. So, this study concludes that in spite of vaccination of breeder flocks at the age of 6 and 8 week and second shot which was given at 16 to 17 week of age, There was high exposure titer, and flocks were infected, exposed to Mycoplasma septicum during the study time period. And this antibody titer recorded were higher than the established post-vaccination baselines. These
Thank you so much, Dr. Sabah Farooq, for presenting today immunomodulatory and growth promoting effects of banana peel powder in broiler chicks experimentally challenged with Salmonella gallinarum. Let's welcome Gulbina Salim.
Areas. We measured interleukin 6 and major histocompatibility class 2 beta growth performance in terms of weight gain, feed conversion ratio, and feed intake, as well as bacterial burden, that is the direct measurement of Salmonella colonization through colony farming unit counts in the tissues. So this is the experimental design. We used 180-day-old broiler chickens. They were divided into 5 treatments, each containing 36 birds with 3 replicate span having 12 birds. The treatment groups included known challenge control, Salmonella gallinarum infected group, 2% banana peel powder group, 4% banana peel powder group, and ciprofloxacin treated group, that is a standard antibiotic against Salmonella gallinarum infection consecutively, respectively. Banana peel powder supplementation from day 3 of the experiment till day 21 of the age of the birds, whereas ciprofloxacin was given from day of challenge for 3 consecutive days. We collected samples at days 7, 10, 14, and 21 to track disease progression and recovery. 3 key tissues, spleen, liver, and cecal tissues, were taken after necropsy, and multiple analysis approaches like gene expression for molecular insight tissue changes were taken. These images show the gross pathological changes in key organs that we examined after infection and showing the classical signs of Salmonella gallinarum disease. The cecum here is swollen and inflamed with that dark mottled appearance instead of healthy pink tissue that is characteristic lesion of Salmonella gallinarum infection. The liver shows here, the liver shows these characteristic spot-like and patches where the infection has already spread systematically. And notice how enlarged and congested the spleen is. It is working over time trying to fight the infection. The banana peel treated groups show markedly reduced changes approaching as compared to challenge. Now, let's look at what's happening at molecular level. We measured 2 key immune genes. First is interleukin 6, which is an alarm bell that alerts the immune system when there is trouble. We found that birds getting banana peel powder had high level of this gene working, which means their immune system were properly recognizing and responding to Salmonella infection. The second gene, MHC class 2 beta, is like the immune system identification system. It helps immune system to recognize and remember foreign invaders. Birds on banana peel powder showed enhanced activity of this gene, meaning that they were better equipped to identify the target of Salmonella bacteria. What particularly encouraging is that we saw these positive immune responses in both liver and intestinal tissues. So the benefits were not limited to just one organ system, and consistently the birds receiving the higher dose of banana peel powder, that is 4%, shows the strongest immune gene activity. This molecular evidence confirms that we saw in the tissue samples Banana peel powder is not just masking symptoms, it's actually strengthening the bird's natural immune defense, helping them fight off infection more effectively on their own. Histopathological analysis in spleen, liver, and ceca is ours that banana peel powder dramatically reduce the immune cell invasion and swelling in the tissues. The inflammation that you can see in infected control group here is much less severe when we added banana peel powder to their diet. Better tissue architecture is preserved, which is really striking is how well these normal tissue structure was maintained. Banana peel powder helped to preserve this normal architecture. All these histopathological images shows that the disease damage was significantly reduced in all three organs, the spleen, liver, and the intestine, instead of severe damage that we see in untreated control. The banana peel powder supplementation showed much milder changes. Here, what really caught our attention was that 4% banana peel powder group was quite similar to antibiotic that is ciprofloxacin treated group. This is quite remarkable for natural supplement. This tells that banana peel powder is providing the same level of protection against the infection as our standard antibiotic treatment. As far as growth performance and bacterial load data is concerned, this is what farmers really care about, the performance and economics. We track two critical measures, how well these birds grow and how effectively they cleared the infection. Starting with the growth performance, birds received banana peel powder at both 2% and 4% level performed just as well as those treated with antibiotics. They gained weight normally, and crucially, they converted feed into body weight more efficiently. This is huge for farmers because feed represent about 70% of production cost, so better feed efficiency directly translate to better profits. What's interesting is that these benefits became stronger over time by days 14 and 21. The advantages were really clear. These suggest that banana peel powder has cumulative effects that build in the bird system. So we actually counted salmonella bacteria in tissues. The birds getting 4% of banana peel powder has significantly fewer bacteria by day 10. They were actively clearing the infection from their bodies, not just by managing the symptoms. The rapid bacterial clearance explains why these birds stayed healthy and kept growing well. We are not just preventing the disease, we are actually helping the birds to eliminate the infection actually. This gives banana peel powder both preservative value for the healthy flocks and therapeutic potential when infection do occur. Conclusion: banana peel powder supplementation modulates key immune related gene expression in terms of interleukin 6 and MHC class 2 beta, improve gut morphology, and enhance growth performance in broilers infected with Salmonella gallinarum. Moreover, banana peel supplementation at 4% markedly reduced pathological lesions in spleen, liver, and intestine comparable to ciprofloxacin. However, these findings highlight that banana peel powder as a promising natural alternative for managing Salmonella gallinarum infection in broiler birds. Thank you for your attention.
Thank you so much, Gulbina Salim, for joining us in this session and sharing your insights with us. Next, we have our 13th presenter. She is also from Government College University, Faisalabad, Nadia Yassin. Nadia Yassin would be presenting pepper nigrum extract supplementation mitigates nickel-induced stress in silver carp. Let's welcome Nadia Yassin.
From the name of Allah, who is most merciful and beneficial, and whose blessings are countless, I have expressed my gratitude of Applied Zoological Society of Pakistan for organizing this outstanding event and give me an opportunity in e-present of 8th International Conference of Applied Zoology 2025. And the title is Black Pepper Seed Extract Mitigates Nickel Toxicity in Silver Carbs. And the scientific name is hypophthalmic thick smolitrix. So first of all, why I have choose this topic is because paper nigrum extract is a natural mitigator of nickel toxicity in fish. Here is the introduction as aquatic ecosystem is facing several type of pollution from industries, agriculture and domestic resources with heavy metals are posing major threats due to their persistence and toxicity. As nickel is a hazard heavy metal that accumulates in aquatic ecosystem through natural and anthropogenic activities, adversely affecting fish growth, physiology, and liver histology. Natural feed additives like paper nigrum possess various types of properties like antioxidant, hepatoprotective, and immunomodulatory properties that may counteract heavy metal toxicity. Hypophthalmic mollitrix is an important aquaculture species, but limited studies have been explored strategies to mitigate nickel-induced stress in this fish. So the aim of this research is to investigate the effect of nickel toxicity on the physiological, biochemical, and histological parameter of hypophthalmic mollitrix and evaluate the mitigating potential of paper nigrum seed extract. So the objective of this research is to assess how paper nigrum supplementation alleviates nickel-induced stress in silver carbs by improving growth performance, hematology, and liver health. Here is the methodology. This is a trial setup where hypophthalmic mollitrix were acclimated for 2 weeks, treated with sodium chloride and kept under optimal water condition. This is a diet design where 6 diets were tested on 216 fishes under 3.6 mg per liter nickel exposure for 60 days. This is the extract preparation where paper nigrum seed were ethanol extracted, filtered, dried, and stored for use. And sodium chloride solution was used for experimentation, and feed formulation was taken where ingredients were mixed with paper nigrum extract and pelletized to maintain a balanced diet. This is a statistical analysis where Bartlett test was performed to confirm homogeneity of variance in the dataset, and one-way ANOVA was applied to analyze growth, hematological, and carcass composition data among treatments. The KHST test was used to identify significant difference between the group that was taken less than 0.05 and coast state software was used for statistical purpose. These are the results. This is a graphical representation of growth performance where nickel exposure severely reduced growth, weight gain, and feed efficiency in hypophthalmic mollitrix. Figure 1 shows the effect of paper nigrum extract on hypophthalmic mollitrix growth under stress condition. Fish that was given 1% paper nigrum extract grew better, almost matching with the control group's results. This is a microscopic image of liver histology of silver carp fed paper nigrum extract against nickel stress at 100 magnification. Figure 1 is a representation of T1 that shows normal central veins and hepatocytes. T2 exhibits epithelium rupture and degenerative hepatocytes, while T3 displays malformed CV, while T4 shows ER and necrosis occur in some cells. T5 exhibits CVD, NC, and HT, while T6 reveals CVD, HT, and ER of the cells. So it is concluded that nickel exposure causes growth retardation, poor nutritional composition, and liver damage in hypophthalmic mollitrix. However, dietary supplementation with 1% paper nigrum extract effectively mitigates these effects, improve growth, hematology, and liver health. Thank you so much. Thank you all of you for your attention. Thank you so much.
It was so much pleasure hearing your thoughts, Miss Nadia. Thank you so much for joining us. Let's welcome Atib Ali from University of Jung, and he would be presenting today efficacy of gingiber officinale and Mantha peperita against Reliantina cysticillus.
Assalamu alaikum. My name is Atib Ali Chishti and I'm from Department of Zoology, University of Chang. Today I'll be presenting my abstract efficacy of Zingiber officinale and Mentha piperita against Relatina cisticelis. This work is carried out under the supervision of Dr. Sidra Abbas and Professor Dr. Arfan Zaman from the College of Veterinary and Animal Sciences, Chang. Now come forward to the introduction. Poultry farming is one of the most important part of our agriculture system. It provides a major source of affordable protein through meat and eggs, and chicken makes up the majority of the production worldwide. But parasitic infections, especially helminth infection, are serious problems. In the poultry, they slows down their growth, reduce feed efficiency, and decrease egg production, leading to the financial losses. One of the parasites, Relatina cisticelis, a tapeworm that infects chickens, it can easily be identified by hooks, suckers, and their body segments. Basically, Relatina genus have three species which infect chickens: Relatina cisticelis, Relatina echinobothridia, and Relatina tetragona. But my area of study is Relatina cisticelis. Farmers usually rely on synthetic and anthelmintic drugs, but the problem is that these drugs can cause resistance and leave chemical residues in the poultry. So my research focused on the exploring natural plant-based alternatives, particularly Zingiber officinale and Mentha piperita, because both have strong long-term history of medicinal uses and known there for safety bioactive compounds. Now, the main goal of study was to evaluate how effective these two herbal extracts are against Relatina cisticelis. For that, I performed both in vitro and in vivo experiments and observed several parameters such as adult mortality test, egg per gram, hematological values, and egg production, body weight, FCR, etc. Now we come forward to the methodology. Dried ginger rhizomes and peppermint leaves were ground and used to prepare their aqueous extract. In the in vitro test, worms were placed in different concentrations for each extract, and their activity was observed over 24 hours to record paralysis and death times. For the in vivo experiments, 75 infected chickens were divided into five groups: one treated with ginger extract, one is peppermint, one with albendazole and other as a albendazole as a synthetic drug, one infected but untreated control group, and one non-infected control group but negative and positive controls. We then measured egg per gram using the McMaster technique, analyzed hematological parameters from blood samples taken from their wing veins, and also recorded egg production, body weight gain, and feed conversion ratio (FCR). Now we come forward our results. First is in vitro results, adult mortality test. In the in vitro study, all treatments affected worm survival, but albendazole produced the fastest paralysis and death of the worms. Both ginger and peppermint extracts also showed clearly activity, through their effects were slightly lower than the synthetic drug. This confirmed that both plant possesses definite anthelmintic potential. Now next, EPG of Relatina cisticelis. When we looked at the egg burden, there was a significant reduction in the number of egg per gram of feces in all treated group, as you shown in the as you see in the graph. When chickens were infected, the EPG were 400 to 450 and 475, but after treated, they were recovered. Again, albendazole showed the greatest reduction, followed by the ginger and then peppermint. Compared to the untreated group, both herbal extracts performed strongly, showing that they can help control tapeworm infection naturally. Next is hematological analysis. The blood profiles of treated bird improved noticeably. Hemoglobin and red blood cells count increased because in infected birds, hemoglobin and red blood cells decreased, and white blood cells level moved back to the normal values. When chicken were infected, white blood cells increased The recovery was most pronounced in the albendazole group, followed by the ginger and then peppermint. These results shows that the extracts not only reduce worms but also improve the bird overall health. When we looked at the egg production, there was a clear improvement after treatment. The highest production was recorded in the albendazole treated birds, while the ginger and peppermint group were showed better laying performance than the infected control. So, as the parasitic load decreased, productivity increased. Now, next is weight evaluation. In terms of body weight gain, the trend was similar. Chickens treated with albendazole gained the most weight, followed by the ginger and peppermint. This improvement reflects better feed utilization and recovery from the infection. Now, FCR. The feed conversion ratio also improved after treatment. Albendazole again gave the best FCR, while ginger and peppermint extract shows a considerably improved compared with the infected control. But where our positive control have negative FCR because we didn't give any treatment to the infected birds. So their FCR continuously decreasing and show you are seeing in they have minus FCR. So overall, although albendazole remained the most effective, both herbal extract perform well and can be considered promising and natural alternatives. Now we next slide. I feel the great pleasure and honor to express my heartiest gratitude to my esteemed supervisor, Dr. Sidra Abbas, and my co-supervisor, Professor Dr. Fanzaman, for their dynamic supervision in designing the research project, keen interest, valuable suggestions, and unwavering support that have been illuminating in shaping my work. Thank you for your attention.
Next, we have Aniba Arshad from Government College University, Faisalabad, and the title of her talk is Investigating the Toxicity of Polylactic Acid Microplastics on the Health and Physiology of Freshwater Fish, Sirhinus miricala.
Bismillahirrahmanirrahim. My name is Aniba Arshad. I am student of MPhil from DC University, Faisalabad. Today I will be presenting on investigating the toxicity of polylactic acid microplastics on the health and physiology of freshwater fish, Sirhinus miricala. Here the introduction. Global plastic production is rapidly increasing with biodegradable polymers like polylactic acid gaining prominence. Polylactic acid, though eco-friendly, degrades faster into microplastics which contaminate aquatic environments. Microplastics originate from industrial waste, textiles, cosmetics, and plastic breakdown, heavily polluting freshwater. In fish, microplastic cause oxidative stress, inflammation, digestive blockages, metabolic disruption, and reduced survival. Microplastic ingested by the fish can transfer toxic chemicals and pathogens through the food chain, posing risk to human health. Next, aim and objective. Aim to examine the toxicity of the biodegradable polylactic acid microplastics on the health and physiology of the Sirhinus miricala. Objectives to assess the effects of polylactic acid microplastic exposure on the growth, body composition, blood profile, nutrient digestibility, nutrient status, and fish health. To evaluate the overall physiological responses and potential health risk of polylactic acid microplastic exposure in freshwater fish. Next, methodology. First of all, experimental setup. A 90-day feeding trial was conducted on the Sirhinus miricala fingerlings that has high 6.38 to 6.92 gram, acclimated in tanks with optimum water quality, pH that have pH 7.5 to 8.5 and temperature 25 to 28 degrees Celsius, and dissolved oxygen is 6 to 7.5 milligram per liter. Next, formulation 6 isonitrogenous and isocaloric diets were prepared with polylactic acid microplastic levels of 0.5%, 1%, 1.52 and 2.5%. Feed preparation ingredients ground to 0.5 millimeter mixed with fish oil and water. Formic acid is added as the digestibility marker. Polylactic acid microplastic solution sprayed on feed pellets. Experimental design. 3 replicates per diet with 15 fingerlings, each total to 70 fishes. Tanks aerated and maintained under controlled conditions. Feeding and sampling. Fish feed at 5% body weight. Tanks cleaned after feeding. Fecal samples collected towards post-feeding bias wells and stored for the analysis. Growth performance was evaluated using food conversion ratio, specific growth rate, and body weight gain in grams and percentage for control and treatment groups. Chemical analysis, hematology, digestibility, hemopathology, minerals contamination tools was observed under the growth performance. This table shows growth study of the Silhanus merigella fish after the exposure of the silk fibrion microplasticals based biodegradable polylactic acid microplastic. Two groups, first control group and second treatment group. In control group, no exposure of the polylactic acid microplastics, and then noted the initial growth rate, body weight, final body weight gain percentage. All of these are noted for control group. Fishes show more growth and optimum growth show at control group because no exposure of the polylactic acid. Next, we show digestibility, microplastic digestibility in feeds, digestibility in feces. We observed microplastic digestibility in feed, then we observed microplastic digestibility in feces. All these all these parameters show more growth in control group because there is no microplastic digestibility in control group. We observe this stable blood profile of fishes Silhanus merigella after exposure of the polylactic acid microplastics. More hemoglobin, more RBC, WBC, PACB show growth at the control group than treatment groups. Next, histopathological study. Histopathological examination revealed that polylactic acid microplastic caused intestinal damage in Silhanus merigella with the high treatment groups, showing severe destructions of the intestinal villi. All treatment groups exhibited varying levels of the villi deformation, while age and staining also indicated serosa damage. Overall, polylactic acid microplastic, particularly at 2.5% concentration, significantly disrupted intestinal structures, impaired digestion, and the nutrient absorptions. This figure shows body compositions of the carried proteins and carried fat, and the second table show ash and moisture content. In conclusion, the finding of this research suggests that Silhanus merigella was negatively impacted by the polylactic acid microplastic exposure throughout a 90-day period. Significant alteration in growth, indices, body composition, nutrient digestibility, and hematology were seen in the treatment groups. Following exposure to polylactic acid microplastic, intestinal histology and mineral composition were also disrupted at a high dose of the polylactic acid microplastic. These findings suggest that prolonged exposure to polylactic acid microplastic may disturb fish health based on our observations. Thank you so much, everyone.
Next, we have Muhammad Danish Latif from US, and he would be presenting today impact of partial substitution of fish meal with lysine supplemented and 5 days pre-treated rice protein concentrate on the growth performance, nutrient digestibility, and enzyme activities of striped catfish. Let's welcome Muhammad Danish Latif.
Assalamu alaikum, everyone. And first of all, I would like to say thank you to the organizing committee of the 8th International Conference on Applied Zoology 2025 at the Punjab University, Lahore, in collaboration with Government College University, Faisalabad. So first of all, as far as my introduction, I am Muhammad Danish Latif from the Department of Fisheries and Aquaculture, UVAS, Lahore. And today I'll be presenting about the impact of partial substitution of fish meal with lysine supplemented and phytase pre-treated rice protein concentrate on the growth performance, nutrient digestibility, and enzyme activities of striped catfish Pangasius hypothalamus. Next. So, introduction. Fish meal is the best animal protein source in the aquafeed industry, but it has problems of haphazard supply and long-term availability. The scarcity of fish meal urging the aquaculturists to explore novel alternative protein sources, such as plant protein sources. However, the plant proteins also possesses some challenges in the aquafeed industry, like anti-nutritional factors (ANFs) presence and amino acid deficiency. Rice or Rhiza sativa is a vital cereal crop around the globe. Though protein concentration in rice is very low, 6 to 7%, but the rice protein concentrate, RPC, comprises more than 65% protein. Moreover, like other plant proteins, RPC also possesses ANFs like phytate and low quantities of lysine. Phytic acid, also known as phytate, is the major storage form of phosphorus in plants. Back. Phosphorus in plants. Fish are unable to hydrolyze phytate, which is the main reason behind the limited utilization of plant proteins in the fish feed. Moreover, lysine deficiency results in decreased digestive, absorptive capability, feed utilization, and growth retardation. Pakistan produced 9 million metric tons rice in year 2023 and 2024, but its byproducts are not that efficiently utilized. The conversion of byproducts to RPC and its utilization in fish feed after treating it with phytase on exogenous enzyme and lysine balancing will provide an alternative nutritionally balanced ingredient. Therefore, the present experiment is designed to study the effects of treated RPC on growth performance, nutrient digestibility, and enzyme activities of Pangasius hypothalamus. Statement of the problem. Due to the scarcity of fish meal in the aquaculture industry, the plant proteins emerge as a compatible candidate due to its easy availability among different substitutes of fish meal. RPC is emerging as the best example of plant proteins, especially in Pakistan due to its availability, lower cost, high crude protein, and low lipid content. But unfortunately, due to lysine deficiency and phytate content, the use of RPC in aquafeed industry is limited. Therefore, these studies aim to overcome these problems by pre-treating RPC with phytase and exogenous incorporation of lysine. Objectives: to assess the effect of partial substitution of fish meal with phytase pre-treated RPC on the growth performance, nutrient digestibility, and enzyme activities of Pangasius hypothalamus. To evaluate the effect of partial substitution of fish meal with lysine supplemented rice protein concentrate on the growth performance, nutrient digestibility, and enzyme activities of Pangasius hypothalamus. To appraise the synergistic effect of partial substitution of fish meal with phytase pre-treated and lysine supplemented RPC on the growth performance, nutrient digestibility, and enzyme activities of Pangasius hypothalamus. Hypothesis: fish meal could be partially replaced with phytase pre-treated and or lysine supplemented RPC with lysine supplementation in the diet of Pangasius hypothalamus without affecting the growth and health status. Materials and methods. Study site. This study was conducted at the Fish Seed Rearing Unit of the Department of Fisheries and Aquaculture, UVAS, Ravi Campus, Pato. Experimental species. The striped catfish Pangasius hypothalamus was used as experimental species. So during the fish husbandry and feeding, so in this slide, you can see the whole fish husbandry and feeding procedure during the whole trial. So formulation of experimental diets, we started with the grinding and then mixing, then pelletizing, shade drying, and packaging in storage for the diet 1, 2, and 4. While for the diet 3 and diet 4, we started with the grinding of the ingredients, phytase addition, mixing, and then incubation at 40 degrees centigrade for 15.5 hours, then drying, then wet ingredients mixing, pelletizing, then shade drying, and packaging and storage for the trial. So in this slide, you can see the formulation of experimental diets, and you can see the composition of experimental diets and their composition as per the percentage dry matter of the experimental diets. So in this slide, you can see the harvesting and sample collection plan of this trial. So these are the formulae for the calculation of growth performance and the nutrient digestibility, in which we calculate the weight gain percentage, SDR, survival rate, feed conversion ratio, protein efficiency ratio, feed intake, survival rate, and ADCs. So these are the lab analysis at the conclusion of trial, which include proximate analysis, serum biochemical indices, intestinal enzyme activities, and intestinal morphometry. So for the statistical analysis, the experimental design was completely randomized and data was evaluated statistically using polynomial contrast. The mean values were compared using orthogonal, polynomial contrast, linear, and quadratic. SAS computer software was used for analyzing the data statistically. So these are the results of growth performance. In this slide, you can see the effect of partial fish meal replacement with phytase pretreated and lysine supplemented rice protein concentrate on the growth performance and the biological indices of the fungaceous hypothalamus fingerlings. In this slide, you can see the effect of partial fish meal replacement with phytase pretreated and lysine supplemented rice protein concentrate on the nutrient digestibility of fungaceous hypothalamus fingerlings. In next slide, you can see the effect of partial fish meal replacement with phytase pretreated and lysine supplemented rice protein concentrate on serum biochemical enzymes of fungaceous hypothalamus fingerlings. In this slide, you can see the effect of partial fish meal replacement with phytase pretreated and lysine supplemented rice protein concentrate on the histomorphometry of fungaceous hypothalamus fingerlings. In this slide, you can see the effect of phytase pretreatment and lysine supplementation in the rice protein concentrate on the digestive enzyme activities of fungaceous hypothalamus. In this slide, you can see the effect of partial fish meal replacement with phytase pretreated and lysine supplemented rice protein concentrate on the body composition of fungaceous hypothalamus fingerlings. So in conclusion, hence it is concluded that fish meal can be partially replaced with lysine added and phytase pretreated rice protein concentrate without affecting the growth performance, nutrient digestibility, digestive enzyme activities, and hepatobiochemical enzyme activity in the strip catfish fungaceous hypothalamus. Thank you for your attention.
Thank you for joining us, Muhammad Danish Latif. Next, we have Miriam Fatima from University of Jang, and she would be presenting today nutritional impact of Britannia coagulans on broiler health.
Assalamualaikum, respected organizer and fellow researcher. I am Miriam Fatima from Department of Zoology, University of Jammu. Today I will be presenting my abstract or research topic, and nutritional impact of Britannia coagulans on broiler health. This study explored the potential of Britannia coagulans on broiler health and potential of a medicinal herb as a natural alternative to antibiotic growth promoter in poultry production. As poultry is the world's fastest growing source of animal protein, it has short rearing cycle and also low production cost. However, overuse of antibiotic growth promoters had led to antimicrobial resistance concern. Hence, researchers are now turning to herbal phytogenic feed additive that can maintain bird performance safely, and that feed additives can be used in bird feed or in poultry production and can be safe for the animals. And among of the one is Thaniocoglin, commonly known as Panidoli. It is rich in bioactive components like retinoids, flavonoids, alkaloids, and tannins. It is recognized for its many properties like antibacterial, antioxidant, hepatoprotective, immuno, immunomodulatory, hepatoprotective properties. But there is limited research or studies exist on its use in broiler feed, which motivated this research. Major objective of this study were to evaluate the effect of ethanocoagulin fruit powder on broiler growth, feed efficiency, feed conversion ratio, to determine its influence on hematological parameter, and finally to assess the carcass characteristics and histomorphological changes in liver and kidney. And next, the material method of the research was the experiment was conducted for 42 days using 308 broilers. Broiler was divided into four groups: T0 control, T1 250 mg per kg, T2 350 mg per kg, and T3 450 mg per kg of ethanocoagulin fruit powder in basal diet. The fruit was clean, powdered, and stored in airtight containers. Both performance the parameter which was observed many and the carcass yield and organ weight were measured at the end of the trial, while histological examination of liver and kidney were performed after staining. Next, the most important section, the results. First of all, we will discuss the results of the growth performance. Uh, Broiler which were supplemented with the ethanocoagulin showed a clear improvement in growth performance. The T3 group, which was fed with a high dose of ethanocoagulin fruit powder, show the highest final body weight and the best FCR in the feed conversion ratio. Growth improved in dose-dependent manner up to 450 mg per kg, which is suggesting enhanced nutrient utilization and metabolism. Next, the bar chart for the weekly body weight and body weight gain, feed intake and feed conversion ratio. These bar chart illustrate the upward trend in body weight and corresponding improvement in FCR. Next were hematological parameter. Dietary supplementation significantly improve hemoglobin, red blood cell count, HCG, MCH, white blood cell, and at the same the optimal result were seen at 350 and 450 mg, indicating that moderate to higher dose of ethanocoagulin positively influence the blood health and immunity. The hematological profile of the chicken were improved in dose-dependent manner, with 3C was most effective. This is a bar chart which shows that all the treatment group and all hematological parameters. Next, we have the result, histomorphology result. The histomorphology results histological observation shows that there was intact structure of kidney and liver in control and T1 group. In liver histomorphology, there was the mild histological alteration was observed uh, in elongated hepatic duct, disrupted hepatic sinusoids, localization, and degenerating nuclei in hepatocytes. And same was the case in the kidney histomorphology. We observed that T2, T0 and T1 control and T1 group, they were normal kidney structure with well-defined glomeruli, elongated renal tube, no visible lesion was observed. But in T2 and T3, mild degenerative intestinal observed, atrophy of glomerulus, visualization and narrowing of renal tubule. The result of histomorphology shows the mild dose-dependent histological alteration and higher supplementation. These are the results which shown the hepatoprotective and nephroprotective changes in the structure of kidney and liver. Then we have the result of carcass. In carcass, the groups which were treated with hernia have showed increase in breast weight and higher yield of breast and thigh meat. The result reflected improved nutrient deposition and muscle development. No major differences was observed in giblet weight. And next is the chart which summarize overall enhancement in carcass yield percentage among all treatment. Next, from all of the research that were conducted, we conclude that the Withania coagulin can enhance the growth, blood health, and carcass quality. In summary, supplementation with Withania coagulin improved broiler growth, blood profile, carcass quality, and protected vital organs. The observed benefits are attributed to bioactive components such as ethanoids, flavonoids that enhance digestion, nutrient absorption, and cellular defense. Therefore, Withania coagulin uh, can be served as promising natural genetic growth promoters in poultry feed, supporting both bird health and sustainable production. I sincerely acknowledge and thankful to my supervisor, Dr. Sitra Abbas from University of Jammu, and my co-supervisor, Dr. Yasir Abbas from Sivas Jammu. I also thankful to my Department of Zoology, University of Jammu, for providing me necessary facilities, and also thankful to ICAZ 2025 organizing committee for this opportunity to share my work. Thank you so much for your attention. I will be happy to have any question or comments. Thank you.
Following Maryam Fatma, now we have Sumaya Jalil from GCW Sialkot. And the title of her talk is Possible Protective Effect of Camphorol Against Triclosan Induced Genotoxicity and Oxidative Stress in Labiorrheta.
Assalamualaikum. First of all, I thanks to the Applied Zoological Society of Pakistan for organizing a conference on applied zoology with the collaboration of University of Punjab, Lahore, and the Government College University, Faisalabad. My name is Sumaya Jalil, and I am from Government College Women University, Sialkot. I'm excited to share my findings today on a critical environmental and toxicological issue, the effect of common antimicrobial agent triclosan on the aquatic life and a potential natural solution to mitigate this harm. We all live on a blue planet where water is the very essence of life. Yet across the globe, our vital aquatic systems, from the largest ocean to the smallest rivers, are under siege. The silent, pervasive crisis of the aquatic pollution is transforming these life-sustaining waters into a toxic soap. It's a crisis defined by everything from massive plastic islands to the invisible threat of molecular contaminants that undermine the health of every organism, including the one we rely on our food. So, while visible pollution like plastic gets a headline here, my presentation focused on a far more insidious threat, the chemical burden on our waterways. Our homes, industries, and farms are continuously discharging a complex mixture of emerging contaminants, including the pharmaceuticals, pesticides, and personal care products. These chemicals may be invisible, but their impact on the aquatic life is profound. This brings us to the prime example, triclosan, which is a ubiquitous antimicrobial agent. This antimicrobial agent enters the aquatic environment primarily through the wastewater discharge and is also known as significant effect on fish even at low environmental relevant concentrations. This is one of the this one becomes one of the most frequently detected organic wastewater contaminant in the world. These are mostly found in the trash bags, toys, soaps, and packaging material, which are one of the common household products. The effects of triclosan on fish are broad, encompassing the acute toxicity, behavioral changes, and the various sublethal impacts, primarily by acting as an endocrine disruptor and inducing the cellular damage. While triclosan possesses a clear threat to aquatic life by inducing the significant oxidative stress, genotoxicity, my research explores a promising natural solution. We have investigated the protective effects of camphorol, a flavonoid commonly found in fruits and vegetables. This compound is renowned for its powerful antioxidant properties, which allow it to directly neutralize the harmful free radicals and boost the fish's natural defense system. The findings demonstrate that when co-administered with triclosan, camphorol acts as a potent mitigator, effectively shielding the fish from the cellular and DNA damage. My specimen is the fish, which is rohu, which is one of the major Indian crops of the South Asia. It contains the nutrients like vitamins, minerals, and including the iron and zinc, which helps the boost the immune health, bone health, cardiac health, etc. My study objectives is to determine the acute toxicity of triclosan of the in the fish of Labiorhita. The second one is to calculate the oxidative stress and genotoxicity, which is induced by the sublethal exposure of triclosan in Labiorhita. And the third one is to evaluate the protective effects of camphorol against the triclosan-induced oxidative stress and genotoxicity in Labiorhita. Then we come to the methodology factor. First, collection and the acclimatization of fish is done. The stock solution is being prepared. The acute and sublethal toxicity is calculated, and the analysis of antioxidant enzymes is done. DNA damage is done, and all the data is calculated through statistical analysis. In the acute toxicity, the LC50 of triclosan is calculated by the probit analysis method. Here, the median of this is 0.0434, which is the LC50 of triclosan. During the sublethal toxicity, the protective effects of camphorol is determined, and the physicochemical parameters are measured on criteria trial. Antioxidant enzymes like CAD, POT, SOD are calculated. These enzymes have the same trend of determination of toxicity, while the T-bars and ROS are the of oxidative stress markers and also have and they both have the same trend. DNA damage is done by direct preparation, lysis of DNA, and the natural electrophoresis process, also known as gel electrophoresis. Statistical analysis is done by using the ANOVA, and in the SPSS software, significance value was set at 0.05. During the process of acute toxicity, the temperature is being constant, pH is being constant, and hardness, while the carbon dioxide, calcium, and ammonia is increased, and the oxygen and magnesium is decreased. These are the graphs of CAD, POT, and SWOT. In all these graphs, toxicant group is decreased, while the camphorol, which has the mitigative agent, is given with the mixture of triclosan, have a reduced activity. These are the bars of ROS and T bars in which the activity of the toxicant is increased while the rest are decreased. Same is the procedure with the CAT, POD, and SOT, which has the same trend of the toxicant group is decreased while the remaining rest is increased. These are the graphs of kidney. Then DNA damage is done. DNA damage is used to check the length of tail, which is the main factor. In in checking the chromatin material which is present in the cells of liver. This is the cell of liver at a 40x magnification. A is the control group, while the remaining are the toxicant with the mitigative. As you can say that B is the toxicity and have the largest tail. These are the graphical representation of head length, tail length, and head DNA, and these are the graphical region of head, tail DNA and tail movement. As you can say that the tail movement in the triclosan group is increased to a significant level, while with the other groups the level is decreased significantly. This is the representation of the cells of the kidney of Leburohita at a 40x magnification. They also have the same results as that of the liver. To conclude this topic, I would like to say that triclosan caused a strong oxidative stress and genotoxicity in different organs of the experimental fish, while the administration of camphorol showed that it has a capability to enhance antioxidant enzymes and protect against the DNA damage in a dose-dependent manner. These are the references, and these are the tabular representation of the antioxidant enzymes with their p-value, and these are the tabular representation of comet analysis with their p-value.
And finally, we have Kamal Shehzadi from GCW Sialkot. The title of her talk is Therapeutic Potential of Camphorol Against Triclosan Induced Histopathology and Hematological Alterations in Leburohita. Let's welcome Kamal Shehzadi.
Assalamualaikum everyone. My name is Kamal Shehzadi and I am a final year MA student from the Department of Zoology, GC Women University Sialkot. I feel really honored to be here as a part of ICAZ-2025 and I'm really excited to present my study here because I think this conference which is regarding the fostering of innovative solutions for food medicine industry. So my research topic was therapeutic potential of camphorol against triclosan-induced histopathology and hematological alterations in Leburohita. So aquatic ecosystem, we consider it being rich in biodiversity because it is home to a lot of aquatic species, and fish is regarded as one of the unique aquatic species. But with that being said, it is prone to many major threats, some of which I've enlisted here include habitat destruction, overexploitation, water pollution, and invasion by exotic species. So in our study, we used triclosan as a toxic agent, which is a broad spectrum antimicrobial agent, and it is used extensively in our daily lives. It is involved, it is present in personal care items, it is present in healthcare items, the personal care items like the mouthwashes, the toothpastes, the antimicrobial soaps, in the healthcare items like the surgical gloves or the antiseptic solutions also have some quantity of triclosan in it, and also triclosan is present in sports items and household items as well. So despite being its broad usage, FDA has banned triclosan as an active ingredient in antimicrobial soaps. But since antimicrobial soaps were not the only thing in which triclosan was present, so ultimately FDA had to approve the quantity of 0.3% triclosan in toothpastes and other items as well, the items of daily use. So today, the most of our daily life items, they can have triclosan in it, but their ranges are mostly from 0.1 to 0.3% of the weight of the product. There have been studies in the past which have explored the toxic effects of triclosan in fish, and these studies explored the effects being endocrine disruption, neurotoxicological effects, behavioral alterations, hematological and histological alterations, and oxidative stress and DNA damage. So moving on, we used flavonoids as a compound to counteract the toxicity caused by triclosan. So flavonoids, basically they are polyphenolic compounds and they are of organic region, means they are plant-based products and they impart fragrance, flavor and color to the compounds. And flavonoids are mostly known for their antioxidant properties because they are rich in antioxidant compounds. And in addition to that, they have many other beneficial properties like being anti-inflammatory, anti-diabetic, anti-cancerous, and cardio and neuroprotective agents. So the specific flavonoid that we employed in our study is camphorol, and as all flavonoids, camphorol is also a naturally occurring flavonoid, which is found in broccoli, strawberries, apples, teas, and coffees, etc. And it was named in the honor of Dr. Camphor, who discovered it. And camphorol today contributes to the 17% of all the flavonoids in human diet. So basically, it is said that of the diet of an average human being per day, there is around 23 milligrams of flavonoids in our diet. So of that 23 milligrams, 17% is usually camphorol. And the model organism that we used in our study was Labiorhita, and we chose this as our organism because of various reasons. First being easy availability, because it is one of the major Indian carbs, so it is cultivated in Pakistan regularly. So it was highly easily available. With that, it is highly adaptable, which means that it does not come under stress easily, and it is tolerant to slight changes in temperature and pH. And also, it is economically really important for us because of its protein content in the muscles, which is really high. So we formulated this hypothesis for our study, which was that the camphorol can be used as a therapeutic agent against triclosan-induced histopathological and hematological alterations. The study objectives included to investigate the acute toxicity like the 96 or LC50 of the triclosan. Then we evaluated the hematological and histological alterations caused by the sublethal toxicity exposure of triclosan, and ultimately we wanted to check the therapeutic potential of camphorol against the sublethal toxicity of triclosan on both the histopathological and hematological alterations in Labiorhita. So in materials and methods, this was the experimental site, which was the research lab of Department of Zoology, GC Women University, Sialkot, and we collected the fish from a local hatchery in Sialkot, and also we got approval of all of this research from the ethical board of the university. So the experimental design included in total 6 groups. The first one was the negative control group, and the second was the positive control group, which was exposed to only the toxic agent, which was triclosan. The third and fourth groups were the combination groups, which had both triclosan and high and low doses of camphorol respectively. And group 5 and 6, they were like positive control. They had high and low dose of camphorol only. So we acclimatized the fish for 1 week prior to exposing it to any of the toxic agent or the mitigative agent. And during that 1 week, the fish was provided with commercial feed, and we used small sample size like 6 to 8 fishes per group. So throughout the experiment, we determined and maintained the water quality parameters, which included temperature, pH, hardness, dissolved oxygen, and carbon dioxide, calcium, and magnesium as well. So during the evaluation of 96 or toxicity or the 96 or LC50 trial, the Labiorhita were exposed to varying quantities of triclosan, and during these 96 hours, we did not provide any sort of feed to the fish, and the mortality of fish was regarded every 24 hours, like 24, 48, 72, and ultimately 96 hours later, and any dead fish regarding this 96-hour trial was removed immediately from the glass aquariums. And then in the second phase of the experiment, when we exposed the fish to sublethal doses of triclosan and the combination groups also, after their dissection, they were subjected to histological and hematological procedures. So histological procedure, as we know, involves the fixation, then dehydration of the tissues with ascending rates of alcohol, flaring of tissues with xylene solution, embedding them in paraffin wax, then their microtomy, which means cutting and slicing of the cells of the tissues into thin sections, then slide preparation, mounting of the sections on the slide, and ultimately the microscopy of the tissues. And then we also evaluated the hematological parameters, which included the white blood cells, red blood cells, hemoglobin, hematocrit, MCV, MCH, and MCHC. So the statistical analysis was performed using SPSS software. The LC50 was calculated using the probit analysis, and then the analysis of hematological alterations was performed using ANOVA, and then we compared the means using post-hoc Tuckey test. And throughout this analysis, the significant value was set as at 0.05. So in the results, when we determined the LC50 of triclosan, it came out to be 0.43 mg per liter of the water, as I've illustrated here in the probability plot for motility as well. So the water quality parameters during the acute toxicity, I've mentioned here, some of them remained constant, like temperature, pH, and water hardness, while some increased, and dissolved oxygen and magnesium, they decreased. And then during the sublethal toxicity trial, where we administered the 1/3 of the LC50 value of triclosan, there were three parameters which remained constant, including temperature, pH, and water hardness, while there were slight variations in the remaining parameters. So these were the result of histological examinations. These are the histological pictomicrographs of gills. So the picture A here represents the control group, and you can see here that the gills are perfectly normal. They have central intact cartilage, primary lamella, from where the secondary lamella are originating. So it is a normal picture of the gill, whereas B was the group which was exposed to toxicity. So here we can see that there is bending of secondary lamella. There is epithelial lifting, epithelial necrosis as well, aneurysm, and there is also fusion of secondary lamella. Group C, which was the combination group, including like triclosan and camphorol low dose, there were some histopathological anomalies like fusion of secondary lamella, bending of secondary lamella, and there was disintegration of primary lamella. Whereas B, E, and F groups, D was like the combination group with triclosan and camphorol high dose, and E and F were the camphorol low dose and camphorol high dose group. They had perfectly normal pictomicrographs, including the cartilage, the primary lamella, and secondary lamella. All of them were intact. The second organ that we considered was the liver. So here, picture A, as I've mentioned, represents the control group. Here, the histology pictures of liver are perfectly normal, H representing the hepatocytes and HN are the hepatic nuclei. B was the group which was exposed to triclosan, so we can see a lot of histopathological alterations like vacuolization, hepatocyte disintegration, macrophage aggregation, necrosis, and pyknotic nuclei. Group C was the group, a combination group with triclosan and camphorol low dose. While the liver has recovered from the toxicity, there were some alterations which included vacuolization, macrophage aggregation, and some pyknotic nuclei. D, E, and F, as was the case with gills, they have perfectly normal histology, just like the control group, with hepatocytes and hepatic nuclei. The next organ we considered was kidney. So these are the pictomicrographs of kidney. Group A again is the control group and is perfectly normal. G representing here glomerulus, which is surrounded by a Bowman's capsule, and there are renal tubules which are perfectly intact. B was the group which was exposed to triclosan, and we can see there is separation of glomerulus and Bowman's capsule. There is renal tubule separation also, and there is some necrosis as well. C is the combination group with low dose, so there are still some histopathological alterations like separation of glomerulus and Bowman's capsule, vacuolization, and renal tubule separation. D, E, and F are the groups with triclosan plus high dose. E is like the camphorol low dose, and F is the camphorol high dose, and these three groups are perfectly fine, comparable to the control group. So next are the hematological parameters. Here the RBCs, hemoglobin, hematocrit, and MCV, all of them decreased in the triclosan exposed group, and they recovered in triclosan low dose and almost returned back to normal in triclosan plus camphorol high dose group. So from these results, we can conclude that triclosan exposure certainly induces histopathological anomalies in the fish, and also it affects the blood parameters. And as we have discovered, the camphorol supplementation counteracts the triclosan-induced damage. So our recommendations in this case are that while triclosan is really important in our daily lives, it is used on daily basis, but there should be some regular monitoring of triclosan levels. In addition to that, the camphorol, which was the remedial agent used in our study, it should be incorporated into the fish feed because the fish in the fresh waters, which has already been exposed to triclosan, it needs to recover from that damage. So we should incorporate camphorol into the fish feed to recover the damage caused by triclosan, and then we should formulate some policies for the safe limits of use of triclosan, and we should raise public awareness where they should be thoughtful, where they should be mindful of the products that they are using, and they should choose those products which have less, at least to like the minimum amount of triclosan in them. So that was all from my side. Thank you so much.
That brings us to the end of session 5. A big thank you to all our presenters for sharing their inspiring research and to our audience for being such an attentive and encouraging group. The talks today clearly showed how zoological science connects health, environment, and innovation, from lab discoveries to the field applications. Thank you once again, and I hope you continue to enjoy the upcoming sessions of the conference.